Assalamu alaikum. This is our third lecture in the hematopoietic-lymphatic system, third lecture in biochemistry. So in this lecture, we'll be talking about hemoglobinopathies. Let's first define what hemoglobinopathies are. So hemoglobinopathies are basically disorders of human hemoglobin. Basically, we have deficiency in the oxygen transport system. Now, these are really serious diseases as a group because they are the most common, and we have 300,000 infants born each year with a hemoglobinopathy, and they account for about 3.4% of deaths in children below 5 years old. So we're talking about something that is quite serious. Now notice here in the map, that's the distribution of hemoglobinopathies around the world, and notice that how these are concentrated in the old world. The Middle East has a significant share of hemoglobinopathies. So this is something serious, and that would include Jordan as well. So we can classify hemoglobinopathies into different types. We have what is known as qualitative abnormalities. So basically we have mutations that result in different structure of hemoglobin that makes it defective in transporting oxygen. So each amino acid has a function in a hemoglobin molecule. So and there is a good chance that one of them would be mutated, and as a result that would again cause the production of a defective hemoglobin. So we have over 700 variants of hemoglobin molecules because each one of them can be mutated again. So these are known as qualitative abnormalities or qualitative hemoglobinopathies. We also have quantitative abnormalities. Basically, the molecule is fine in structure except that there are less quantities of hemoglobin molecule, whether alpha chain or beta chain, in cells. And these are collectively known as thalassemias. Now, Jordan has a high prevalence of thalassemias. Now, we have a third type of a hemoglobinopathy, and it's known as hereditary persistence of fetal hemoglobin, where basically there is no switching from gamma to beta globin. So we'll talk about this at the end. So let's start with qualitative abnormalities. So basically we're talking about single point mutations, mainly single point mutations. Now these mutations can take place anywhere in the hemoglobin molecule. So they can occur on the surface or they can be internal. Now if they occur on the surface, usually they are asymptomatic. An example is HBE, and we'll talk about it in a few minutes. Now an exception is HBS, or sickle cell anemia. This is a single point mutation that really causes dramatic effect on the function of hemoglobin molecule. Now mutations can also occur internally. Now remember, hemoglobin is a globular protein, so you have hydrophilic amino acids on the outside and you have hydrophobic amino acids inside. Now if this environment is changed, then that would cause the production of an unstable hemoglobin molecule. An example is hemoglobin Hammersmith. Now you can also have mutations stabilizing the methemoglobin, which is basically a hemoglobin with iron in the ferric state rather than the ferrous state. Usually that results in cyanosis, since this hemoglobin cannot bind to oxygen. Now, you also have common mutations in the alpha-1 beta-2 contacts. Now, that would cause an alteration in the equilibrium of the T hemoglobin and R hemoglobin. And that would cause, again, altered oxygen affinity, which is a condition known as polycythemia. Now, let's start with sickle cell anemia, which is basically the most common type of hemoglobinopathies. Now, this is caused as a result of a change in one single amino acid at the 6th position of the beta globin. So, glutamic acid is changed to valine. Now, this is a negatively charged amino acid. It changes to a nonpolar amino acid. Now, this hemoglobin is designated as alpha-2 beta S2 or HbS rather than HbA, where A stands for the adult, if you remember. So what happens here, now this is what a group of hemoglobin molecules look like. So you have single molecules separated and from each other and independent from each other. Now what happens in HbS is that hemoglobin molecules would clump together. They would aggregate, forming sort of like a chain of molecules. Now, this hemoglobin molecule is deficient in binding to oxygen, and it would destabilize the red blood cell as a whole, reducing its half-life from 120 days to less than 20 days. So what happens here is that with repeated cycles of oxygenation, deoxygenation, that would cause clumping and clustering of the hemoglobin molecules. Okay, now it would look something like this. So you have an array of molecules clustered to each other. Now that would cause clumping into capillaries, clogging capillaries, so that blood flow is restricted, causing hypoxia, low level of oxygen in tissues. Now, eventually that would lead to organ damage, particularly in kidneys, heart, and lungs. So the question now is, why is it that hemoglobin molecules or the sickled hemoglobin cluster together? See, what happens here is that right here you have a hemoglobin molecule that is oxygenated. It looks like this. Okay. Now, if it is deoxygenated, it would cause the formation of a hydrophobic pocket within the molecule itself, within the beta-1 chain. Okay, so in a sickled hemoglobin, you have the formation of a valine protrusion or a hydrophobic protrusion that includes a valine molecule. Now, this is the sickled hemoglobin right here. So when it becomes deoxygenated, this protrusion still exists, but you have the hydrophobic pocket in here. So what happens is that this protrusion would fit into this, of one molecule would fit into the pocket of another molecule. So here you have the hydrophobic pocket right here. That's the protrusion, and the protrusion would fit into the hydrophobic pocket. So right here you have the valine protrusion, and it gets inserted into the hydrophobic pocket. Okay, so that would cause clumping of the hemoglobin molecule. So remember that I said that sickling increases with repeated oxygenation deoxygenation cycles. So there are certain variables that increase sickling. One of them is decreased oxygen pressure, which means that you have a high probability of having a molecule that looks like this. Okay, same thing for the other factors. So you have increased the pressure of CO2, decreased pH. That would also increase the formation of the T structure of hemoglobin. Increased 2,3-bisphosphoglycerate, same thing, stabilizing the T structure, and dehydration as well. Now, there is something known as sickle cell trait, which is basically individuals that are heterozygous, meaning that they have both HbA and HbS. This is a case where, again, one allele is normal, it's HBA, producing normal beta chain, and the other one is mutated so that you have the formation of HBS. So you have a combination of HBA and HBS. So this is a normal red blood cell, for example. This is a cell that contains high quantity of HBS. Okay. Now, so these individuals actually do have an advantage. Okay. Now, the advantage is that individuals who are heterozygotes, who have a sickle cell trait, is that they become resistant to infection by malaria. The reason is that you have one reason is that you have lower half-life of red blood cells, so that they are less than they're about 40 days or even less than that. Now, the half-life or the reproductive duration of malaria is about 60 days. So, and malaria infects red blood cells. So that would decrease. So having lower half-life of red blood cells would would prevent malaria from completing its growth inside cells. So these individuals are resistant to malarial infection. And if you look at the distribution of sickle cell allele in Africa, you can see that it actually matches the distribution of malaria in these countries. So it fits. So there's an advantage, a genetic advantage for these individuals. So another qualitative hemoglobinopathy is known as HBC, where basically you have a single point mutation that changes the glutamate at the 6th position, which is exactly the same as sickle cell anemia or HBS, except that it does not change to valine, it changes to lysine. And this beta globin is known as beta C. Now, this hemoglobin is less soluble than HbA, so what happens is that it crystallizes in red blood cells, reducing the ability of red blood cells to change shape as they squeeze in capillaries. Okay. So, for some reason, HBC leads to water loss from cells, leading to higher hemoglobin concentration again in these cells. Now, remember how I said that changing the amino acids on the surface of the hemoglobin molecule it has insignificant effect on the function of the protein? Well, that's exactly the case here. So, contrary to HBS or sickle cell anemia, having HBC causes only a minor hemolytic disorder. So if you look at the previous slide, actually you can see the abnormality in the morphology of red blood cells in individuals having HBC. Now there's another condition known as HBSC. Now these individuals have two alleles with different mutations. One allele is actually a beta S, and the other allele is a beta C. Now, so they produce defective hemoglobin molecule. Now, just like normal individuals or individuals with sickle cell trait, these individuals have mild clinical abnormalities, hardly any clinical consequences, but it is clinically variable. Now, there's another condition known as hemoglobin E. Now, this condition is common in Southeast Asia. So, if someone has a servant, a housemaid coming from Southeast Asia, there is a good chance that this person would have HBE. Now, these individuals they do have a single point mutation in the globin molecule in beta globin, except that now the change is actually on position 26. That would change glutamic acid into lysine. Now, there are two consequences of having this mutation. The first one is genetic. So basically what happens is that there is that would create an alternative RNA splice site leading to the production of a defective protein. Now this protein also, once formed, it's not as stable, of course, as the normal protein, and it would tend to be degraded. So individuals, not only that they have deficient ability of hemoglobin to carry oxygen, they also have lower amount of the beta globin protein. So this condition has both quantitative and qualitative characteristics. Another condition is known as HB Hammersmith. Now, here in this condition, we have a point mutation that changes a phenylalanine into a serine. Now, why is that important? Because this phenylalanine exists in the heme pocket. So it interacts via hydrophobic interactions with the heme molecule, stabilizing the positioning and the interaction of heme into the positioning of heme into the hydrophobic pocket of hemoglobin molecule. Now, if this phenylalanine changes to serine, that would cause destabilization of the interaction between heme and the hemoglobin molecule. That would cause unstable hemoglobin molecule, denaturation of the globin protein, inability of the globin molecule or hemoglobin molecule to bind oxygen, and that would cause ultimately in cyanosis. You can also have mutations that cause stabilization of the T state or the R state. So there is a change in the equilibrium between the two states of the hemoglobin molecule. Now remember that in the T state you have this interaction between histidine with both aspartate, okay, and lysine in the alpha chain, and this aspartate and histidines exist in the beta chain. Now that stabilizes the T state. Now once you have oxygen formation and hemoglobin changes to the R state, you have sliding of the alpha chain, and you have loss of interaction between histidine with aspartate and histidine with lysine. Rather, you have interaction between histidine with aspartate on the alpha chain. Okay, so what happens here is that you can have mutations that can stabilize the T state. That's a condition known as HB Kansas, where asparagine changes to threonine. So rather than having this interaction that stabilizes the R state, now this asparagine is gone, so there's no more interaction, and what you have in here is that stabilization of the T state. Now, contrary to that, you have a condition known as HB Yakima, where you have stabilization of the R state that results in a change of aspartate, this aspartate, to histidine. So you don't have this interaction that stabilizes the T state. Rather, the equilibrium changes to the formation of the R state. So in HB Yakima, you have stabilization of the R state. Okay. So these are some examples of qualitative hemoglobinopathies. Now remember, there are 700 variants, so we can talk about every single amino acid, and there are different names for different hemoglobin variants. Let's talk about another condition or consequence of qualitative abnormalities, which is known as altered oxygen transport. So we talked about methemoglobin briefly, which is basically a hemoglobin molecule that is bound to iron in the ferric state rather than the ferrous state. Now remember that this hemoglobin molecule, which is known as methemoglobin, is not able to bind oxygen. Now, do normal people have methemoglobin? Yes, they do, but the quantity is very low. The reason is that, well, there are two reasons. The first reason is that the hydrophobic pocket surrounding the heme molecule prevents the oxidation of iron from the ferrous to the ferric state. Okay, so what happens is that you have electron rearrangement between the different amino acid residues, preventing this oxidation from taking place. But the other reason is that individuals have what is known as a methemoglobin reductase. See, what happens is that if you have the formation of a methemoglobin, immediately this reductase, the methemoglobin reductase, what it does is that it immediately reduces hemoglobin methemoglobin to a hemoglobin molecule with iron in the ferrous state. This requires cytochrome containing cytochrome molecule within the enzyme itself, and that requires NADH as well. Okay, so you have oxidation of NADH and transfer of electrons to eventually to iron. Okay, this NADH, this reaction takes place, by the way, in red blood cells, of course. Okay, and this NADH, the main source of NADH is glycolysis, and we will talk about this in the next lecture. So there's a condition known as methemoglobinemia, and in people with this condition would have their blood looking like chocolate brown, and they have chocolate lips, okay, because of cyanosis. Now, how can these individuals be treated? Well, first, so why is it that individuals would have a methemoglobin or methemoglobinemia? Well, there are different types of mutations that can take place. So you can have mutations in the alpha chain or the beta chain. So examples include HB Boston. Now in this case, you have mutation in distal histidine. Okay, this is the histidine that stabilizes the oxygen binding to the heme molecule. It's mutated to a tyrosine. So what happens is that that would result in oxidation of iron from the ferrous to the ferric state. Okay, and it attracts water into the pocket. You have another condition known as HBM Iwate, which is a Japanese condition, or let's say that it was discovered in Japan, where you have a mutation of proximal histidine that binds to iron in the fifth coordinate. This proximal histidine is replaced by a tyrosine. Again, it destabilizes or it results in oxidation of the iron molecule. Individuals can also have deficiency in the reductase enzyme, or there are certain drugs or drinking water containing nitrates that would increase the probability of oxidizing iron from Fe+2 to Fe+3. So, as I said, normally what happens is that NADH from glycolysis is the main source for electrons that would be donated ultimately to iron +3, converting into Fe+2. Okay, so an alternative pathway can be used utilizing methylene blue. Now, there are two major metabolic pathways in red blood cells. One of them is glycolysis, and the other one is pentose phosphate pathway. So what happens with the pentose phosphate pathway is that you have the production of NADPH. Now, NADPH is the source of electrons that would be used to reduce methylene blue to another product known as leucomethylene blue. Now, leucomethylene blue can be used as a source of electrons for methemoglobin, resulting in the production of the normal globin molecule. So methylene blue can be used in treating people with methemoglobinemia. So let's discuss now quantitative hemoglobinopathies, which again they're known as thalassemias. So thalassemias are the most common human single gene disorder. They are quite prevalent in the old world, including the Middle East. And basically these are caused as a result of disruption in the alpha to beta ratio. So in red blood cells, what you have is equal amount of alpha chains and beta chains. So in thalassemias, you would have either more alpha than beta or more beta than alpha. Let's start with the alpha-thalassemia. So alpha-thalassemia means that there's underproduction of the alpha globin chains. So basically you have more beta than alpha. Now remember that the alpha chain is produced at the end of the embryonic stage. Okay, so it starts early on and it continues throughout life. On the other hand, the beta chain starts to be produced slowly in the early fetal stage, but there's a big jump right before birth. And it continues throughout life again, having again equal quantities of alpha and beta. Now, if there is low production of the alpha chain, this means that there is a high probability that you will have a hemoglobin molecule that is composed of 4 chains of beta. Such molecule is known as HBH. So an HBH is a hemoglobin molecule that is a homotetramer of beta. Now HBH molecule has reduced oxygen carrying capacity. The main reason for why a person would have alpha thalassemia is deletion mutations rather than single point mutations. Now remember that we have 4 alpha genes. We have 2 on each chromosome. So we have a total of 4 alpha genes. So as a result, normal individuals would have all 4 of them being active. There is a probability that one of them would be mutated. So you would have 3 genes that are active, or you can have 2 that are deleted, or 3, or all 4. That would result in variable severity. So for example, you can have people with only 1 gene deleted. These are known as silent carriers. Okay, so they do not complain from anything. Now, if 2 are mutated, this is known as a alpha thalassemia minor. If you have 3, then this is known as HBH disease. That's because individuals would have a high quantity of HBH hemoglobin, and it becomes very severe if all 4 alpha genes are deleted. So if all 4 of them are deleted, then this is known as Hydrops fetalis. So in this case, you would have high quantity of actually a homotetramer of gamma chains. Okay, so such hemoglobin is known as HB-Bart. Now HB-Bart has no oxygen carrying capacity, resulting in a fetus that is basically dead. So basically you would have stillbirth or death shortly after birth. Now, if 3 alpha alleles are deleted and only one of them is active, then this is known as HBH disease. And the reason is that you have such individuals would have a high quantity of the HBH hemoglobin molecule that is made of a homotetramer, that is a homotetramer of beta. So this is known as HBH disease. This disease is not fatal, but it is symptomatic. Now you have minor alpha-thalassemia, where basically two genes are active. Now these individuals are asymptomatic. And you have a silent carrier where one gene is deleted. These people are totally asymptomatic. So these are again the different possibilities. Normal individuals have all four alleles being active. Here you have in silent carriers, you have three of them being active. And thalassemia trait is when two of them are active. So you can have deletion of the alpha alleles on one chromosome or one from each chromosome. So this is known as thalassemia minor or thalassemia trait. Now the HbH disease, people have mild to moderate symptoms, and in Hydrops fetalis, you have, which is known as thalassemia major, you have all four of them being deleted. So how about beta-thalassemias? Basically, in this condition, results from defective beta-globin genes. Now remember, normal individuals have two alleles, one on each chromosome. So mutations basically result from are basically point mutations rather than deletion mutations. So you can have different types of deletions within the gene itself, the coding region that codes for proteins, or it can be in the translation initiation codon, the splicing condition, splicing positions, or polyadenylation termination signal. Now that would result in the formation of a alpha-globin homotetramer, globin. Now this protein is extremely insoluble, and that would result in destruction of red blood cells. Now, there are different types of beta-thalassemias depending on the severity of the mutation. So if there is complete lack of HbA, that would result in what is known as beta-not thalassemia or beta-thalassemia major. That is, there is no production of the beta globin at all. Now these individuals do suffer from anemia beginning from the first year. These individuals would need transfusions, blood transfusions. The problem with blood transfusions is that there would be accumulation of iron in organs, and we'll talk about metabolism of iron at the end of this series of biochemistry lectures, and that would cause tissue damage and organ damage, particularly liver and pancreas. Eventually that would lead to death in the early 20s. You can also have individuals with beta-thalassemia minor, meaning that there is some production of the beta globin. These individuals are heterozygous for beta-thalassemia. Okay. Now, they have one normal beta globin gene or two that produce low amount of beta globin. Now, usually these individuals are asymptomatic. So there are different types of beta-thalassemias. So normal individuals have normal alleles of beta globin on both chromosomes. Now you can have one of them normal, but the other one can be mutated or defective. This is known as beta-thalassemia trait or thalassemia minor. These individuals are asymptomatic. Now you can have different combinations as well. So you can have both alleles producing little amount of beta globin. You can have one of them being mutated altogether, not producing any beta globin at all, and the other one is defective, producing some. You can have individuals with HBE, okay, but the other allele is mutated, producing little amount of normal beta globin, or you can have these individuals as well. So this is known as beta-thalassemia intermedia. They have variable severity, they have mild to moderate anemia, and so on. But they have iron overload. Or you can have both of these alleles mutated, producing very little or producing no beta globin at all, and this is known as beta-thalassemia major. These individuals have severe anemia. They require transfusion frequently, resulting in what is known as iron overload. Now, the final condition is known as hereditary persistence of fetal hemoglobin. Now, in this case, these individuals have an abnormality in not transitioning from gamma to beta during development. Okay? That would result in individuals producing HbF throughout their life. Now, these individuals do not know that they have an abnormality whatsoever because HbF is still functional and it's good. Now, yes, HbF can bind to oxygen with higher affinity than HbA, but that doesn't really give an advantage to these individuals. Now, the beauty of this, remember when we talked about genetics of the globin synthesis? Well, and I told you, I gave you a hint that this can be used as a treatment for beta-thalassemia. Well, individuals who do not produce any beta globin, why don't we let them produce gamma globin? Okay, so there is an idea of switching on the fetal globin gene in case the beta globin gene is not functional. So there's talk about that. This is taken from a good journal known as Nature Genetics in 2018. Okay, so let's talk about diagnosing hemoglobinopathies. This can be done using electrophoresis. We talked about this before in our biochemistry lectures, and we said that this is a technique whereby molecules such as proteins can be separated according to size, they can be separated according to charge, and so on. So what happens here is that the same thing can be done with hemoglobin molecules, especially those that have single point mutations whereby you have a change of one amino acid to the other. This is particularly true if the amino acid that is changed or that is added would change the net charge of the molecule. So changing glutamate, a negatively charged amino acid, to a valine or to a lysine would change the overall charge of the whole molecule. And as a result, hemoglobin molecules would travel differently. Okay, so this electrophoresis technique depends not on the size of the hemoglobin molecule, rather it depends on the charge of the molecule. So you can read this on your own. Let's look at an example of an electrophoresis result. So basically what you have in here is different samples. In number one, lane number one, we have a standard, meaning that, same thing with number 5, meaning that it's a sample that contains all forms of hemoglobin. So you have HbA, HbF, HbS, and HbC. Now you can see the migration pattern is different among the different hemoglobin molecules. So you have the HbC, for example, traveling close to the cathode, negative charge, and HbS is migrating a bit slower than HbC, and so on. So why is it the case? Why is it that you have this different migration pattern? Well, remember with C, what you have is a mutation of glutamate to lysine. So you have a loss of one negative charge and an addition of one positive charge, and that's why it travels closer to the cathode. Now here you have, with HbS, you have loss of a negative charge, glutamate, to valine, okay, which is no charge. Same thing, the molecule will travel away from the anode and closer to the cathode. Now HbF as a whole molecule has it's more positively charged than HbA, the adult hemoglobin. So these are that's hemoglobin or samples taken from different individuals. You can see that this individual, for example, has only HbA. So this is an adult individual. Look at this individual right here. Sample number 3 is taken from a person having both HbA and HbF with more HbF than HbA. So this is an individual who is a neonate, most probably. Sample 4, HbS only. So this is someone with sickle cell anemia. Look at number 6. You have both HbA and HbS. So this is someone with sickle cell trait. This is number 7, an individual with HbSC. And individual 8 is someone with with sickle cell sickle cell trait as well. So that's how it looks like.